some general do's and don'ts for these kind of workarounds if this is what students you're interested in or parents your child is going to be interested in. There's some biology experiments that can realistically and safely be done at home or in local school settings. One of the most powerful tools for student projects is the use of model systems. So again, a model system is a simpler biological system that helps us understand complex biology. So again, for example, you can use yeast for studying metabolism, stress responses, and microbial growth. You can also use different plants that are commercially and publicly available. You can also look at different kind of environmental factors, things that don't have strict restrictions and regulations or privacy considerations with them. So again, some general do's. You can use food safe or household organisms. This is regulated especially for middle schoolers, and within your science fair specifically. So if you have questions, ask us, or you can also ask the people who run your science fair. Another good do always is your PPE or your personal protective equipment. So wearing gloves, goggles, labeling everything. And then, as Gopi mentioned earlier, you really want to include your controls, repeat your experiments. So the minimum for repeating science experiments would be 3 technical replicates and ideally 3 biological replicates. So that's a total of like 9 different situations as you keep going through everything. And then you also want to track data carefully, and especially for these presentations and posters and projects that students are doing, take pictures as you're working with this different equipment and really document as much as you can, because as Gopi said earlier, one of the best kind of shows that you have a good project is if you write it well enough and you study correctly enough using the scientific method that someone could read your paper and replicate it doing the exact same things that you did. So that's a test of good science. Some major don'ts for safety, privacy, health, all of those reasons that you kind of want to prevent is don't culture unknown bacteria. Not a good idea. There's lots of entertaining things out there, so you don't want to get exposed to something that can make you sick. Do not use very regulated human or animal samples unless it's within a lab under PI, under supervision, that you have a mentor directly for a wet lab. Again, we can use those model systems, but you don't want to use like HIPAA regulated human samples.